Hello everyone, my name is Victor. Today I'm going to tell you about the effect of Asian dust input on eukaryotic phytoplankton community structure in the open area in the northwestern Pacific Ocean. Before going to the topic, I will introduce some words. Planktons means very small plant and animal or other creature that float on the surface of the sea, and it can be divided into the three following portions. First is bacterial plankton. It is component by bacterial plankton. And the second is zooplankton. It is component by animal plankton. And the third is phytoplankton. It is component by plant plankton. And today we only talk about the phytoplankton. And northwest Pacific Ocean. In the spring, strong west wind can carry an amount of 64 kilogram Asian dust in this area, and the flower feature was this phenomenon. And then seasonal physical and chemical environment was insignificant. It means the weather here is constant and it will be no special change. And then water from the depth is not transferred to the surface layer, so the water layer can remain stable. Now we start to talk about topic. How do dust affect phytoplankton? The dust generally components by atmospheric source such as sandy winds or eruption, volcano eruption, or pollution. And it also component by the nutrient in the micronutrient scale, such as nitrogen, phosphorus, and silicon. It can provide the need of phytoplankton's growth. And in the micronutrient scale, such as ferrous, copper, and zinc, these elements are poison to the phytoplankton. So the research purpose is to study the effect of Asian dust on growth and community structure of the phytoplankton in northwestern Pacific Ocean. And the second reason is that many previous studies use chemical analysis or hypothetical model. It is not direct evidence. Hypothetical means it is suggest or image that is not real or true. So the sample size, we choose the station as 26 and 136 longitude and latitude, which is in the oligotrophic and nutrient region. Oligotrophic means this is lack of nutrient. And here is the sample size. And then the dust we used in experiments was derived from the MUS lab here. And then we used golfer bottle mount on the zebra city assembly to collect the surveyed seawater at 5 meters deep. And then we filter the water with 200 micrometers into 3 groups of bottle. The first is controlled. This is no dust added. And the first one is 4 milligrams Asian dust added. And the dust is 40 milligrams Asian dust added. And then we put these 3 bottles into 20 liters seawater, and the temperature of the system was controlled by circulating water. And also, we covered by glass. The system was also covered by glass to reduce 30% of light intensity correspond at 5 meters. And then the bottle will open and shake at 5:11:17 o'clock at Beijing time to ensure enough dissolved oxygen and to prevent phytoplankton aggregation. Aggregation means these be together. And then we observing 3 bottles in next 7 days of phytoplankton growth and doing data processing. First is environmental. We can see the temperature is between 25 and 17 range, and the latency have the maximum value in the light density have the maximum value at the second days, and the other density is between 0 and 400 range. And the nutrient, it has the 4 part of nutrient. And if the graph have the star mark like this, it means that there was an extremely significant between the three samples. So we can see only the NO3 minus in dust 2 was significantly higher than in control in dust 1, and also it consumes more. And then we talk about chlorophyll A. The chlorophyll A can color by the microphytoplankton and nanophytoplankton and picophytoplankton. And chlorophyll A is an important element that the phytoplankton doing light operation. And we can separate from the two parts. So we can see that the macro and nanophytoplankton was significantly higher than the dust 1 in control and the total source. And then we talk about the phytoplankton. There are the 4 dominant groups. We can see the four groups are all increased, but didn't differ significantly among the 3 samples. And then we are talking about the non-dominant group. And we can see that both of them are decreased but didn't differ significantly among the 3 samples. And then we talk about the variation in the abundance and the species number. It's not to let that the word abundance means is not the diversity. It will be confused. And we can note that the abundance is increased by days and the species number is decreased by day. Now discussion on the chlorophyll A figure and the abundance figure. We can see that the chlorophyll A is increased by time and correspond to the abundance. It proves that the dust added input will increase the productivity in the Northwest Pacific Ocean. And then on the figure we can know that after the dust added, NO3 minus increased considerably more than others, indicate that the increased fundamental growth was caused by plant limitation. And then we can see the 4 dominant groups and non-dominant group. The 4 dominant group is increased by time and the non-dominant group is decreased by time and even disappear. And correspond to the species number, we can see the species number is declined by time. So the diversity was decreased. And then we talk about structural change. From hierarchical cluster analysis, we can transfer this to the multiple dimension scale. The multiple dimension scale, we can see all the species in the one circle. And if the species is reaches, and it should be separated by the main circle like this. And this region is all in almost in the same circle. It means that the region here, species is become single and the diversity is declined. And conclusion, we know that Asia does cause more productivity, and Asia does change the structure of the Northwest Pacific Ocean, and Asia does make diversity decline. And that's all. Thank you.
Why the iron is poisonous element to phytoplankton? Because I think iron can make the phytoplankton bloom.
You want to know why the iron mental, you want to know the mental element will cause the phytoplankton bloom?
I mean, why you say the poisonous element like iron or what blah blah blah? Why?
You said this feature why the mental why iron element will poison to the phytoplankton. The research said the ultimatum will cause the phytoplankton change his cells in his I don't know how to say change his interior cells in his part, so it can maybe cause the nutrition can be difficult, so it can cause the poison to the phytoplankton.
Do you know what's the difference between chlorophyll A and B?
You want to know the difference between the chlorophyll A and B, and I don't know how to define the chlorophyll A and B, and because the research only said chlorophyll A, so.
Result has 4 parts. I want to know which parts be affected by those affect phytoplankton is more bigger.
You say the list 4 parts, which is the more important to the phytoplankton. The picture shows that the chlorophyll minus is more important to the photophytoplankton.
What did you say, sorry?
The chlorophyll minus this nutrition element it can influence the phytoplankton more.
Environment, nutrient, and chlorophyll A. This. Repeat your question. Okay. Your result has 4 parts.
Yeah.
And I want to know which parts be affected by thus affect phytoplankton is more.
You want to know which part is more important to the phytoplankton?
More affected by dust.
More by dust. Because the dust is composed most of by nutrients, so I guess it is more important by nutrients.
Can you explain why the ancient dust caused the phytoplankton decrease?
You want to know why the dust what?
Ancient dust caused the phytoplankton decrease.
You want to know why the dust caused the what decrease?
Phytoplankton.
The pictures show that the phytoplankton abundance is increased because the chlorophyll A is increased, so the correspond to the abundance, so that the abundance is increased.
I want to ask why the phytoplankton diversity is decreasing.
You want to know why the phytoplankton diversity is decreased, and because the picture shows that the dominant group will increase, but the non-dominant group will decrease and even disappear. So the species number will decline by time.
There has many star-shaped symbol in your graph. What's that mean?
Can you say it again?
There has many star-shaped.
So star.
Like that.
You want to know what is the star shape means in the figure? The star shape in the figure is that there are extremely significant between the control and dust 2 sample. And this is a statistics way to get it.
Okay, so I think Victor, he gave a clear talk, but I think in my opinion, you spoke too fast. Considering that your talk was below 10 minutes, I would say you can simply make 10 minutes by slowing down your talk speed and make it easier for the audience to follow you. Because here you have figures like this, and each figure has actually 4 panels. So and each panel has 3 lines. So I was actually quite busy really figuring out what's going on here. And this is somehow related to the way you presented your figure. I noticed that even for this very first figure, you did not really introduce them. You have to say, you have to say this is day of what? Not just days. Days of what? Of incubation temperature, and the light intensity. And then later on, here you have many lines. And I think this should follow one of your questions. You have to introduce what this means, because these are the most notable features in the figure. I think it won't be too difficult to say the stars means statistically significant. I think most of the students here can understand. However, I don't remember if you have introduced all the lines here. At least I was really figuring out, okay, what's the black and so on. So you have to really take time to introduce the pattern. And furthermore, please do not expect that the audience read the graph and they can immediately follow you to the concluding remark. I think it's really nice that you print the concluding remark right next to the figure. However, what was missing was you did not take your time to introduce the pattern of the data. For example, an easy way to do that is just use your pointer and say, look at this, the black line increase over time, and the stars means they are statistically higher than the other 2 groups. You have to use your time to introduce the pattern of the data before you draw the conclusion. What was good for your discussion is you have this structure of a separate discussion and results and discussion. However, in your discussion, you use this small plus or like a thumbnail of your figures to remind us again the key features. So that was really nice. Because you have so many figures, I would suggest that if possible, try to, for example, just put this immediately on the figure. Help us to understand what's really going on. And usually you don't need to say A or B, it's only for paper. For presentation, you just say this is for this cryptophyta and that is for chrysophyta, etc. And you can see that your result in the discussion is a little bit black and white. So for example, if you say cryptophyta, you can give us more feeling by adding an image of this creature. So we have a better feeling what feature you are talking about. Okay. And the conclusion was really nice because compared to many conclusions I have seen so far, this one is relatively specific. But still you can improve by saying more regarding what you mean by change the structure. So tell us a little bit more. Yeah, and these 2 are really specific. That was nice. Okay, and therefore another suggestion is I think you can reduce the details of the methods, say less about the method, and talk in results and discussion. All right. And another thing is your multivariate results. I don't know how many students here know multivariate statistics. This one is actually quite difficult, and if you really, my suggestion is either you remove this or you take time to explain them well. For example, when you were explaining this, if I were you, I would really make it this big, and take time to say what's this, and what's this, and how do you read the graph. That's how you should introduce, the way to introduce the statistical figure. And if you think this graph gives you a better idea, you can just focus on this one. However, you have to make this one really big and tell us what, because you see this graph doesn't have any x and y axis, what's going on, how to read this. And you have these lines and that spots. So it's actually a complicated figure. Take time to explain it if you want to show it. Okay, so yeah, I think you have all the content, but next time you have to check the way you introduce the data. Okay? Okay, let's thank Victor.